Good morning, everybody. My name is Annapurna. I'm from Sri Krishna Arts and Science College. The title of my paper is to find a potential inhibitor against UMP CMP kinase in Albugo candida. Albugo candida is nothing but a plant pathogen that infects the Brassicaceae species, that is the mustard crops, cauliflower, cabbage, and the turnip, to cause a disease called white blister rust. These candida species are fungus-like microorganisms that belongs to oomycetes. They infect the plant in the seedling stage, growing stage, flowering stage, and also the fruiting stage. And different races of candida infect different plant species. The white rust infects the stem, leaves, and the inflorescence. On the outside, it forms white colored crystals of 2 millimeter in diameter packed with sporangia. In the inside, it forms abnormal growth forms and also sterile inflorescence. The sporangia spread by wind, water, and insects to other plants, causing the disease. We can see I've put two plants infected with Albugo candida species. We can see the pistils here. And UMP CMP kinase, I've selected this enzyme because there are totally 19 enzymes that are responsible for the infection, and we are totally not sure about which is the enzyme that's totally responsible for the infection. And UMP CMP kinase is involved in various biological processes like ATP binding and pyrimidine nucleoside synthesis. It also acts as a transferase. And methods is Galaxy CO Lab. I've used this web server to predict the structure of the protein using template based modeling, and Ramachandran plot, the protein structure obtained from Galaxy CO Lab is validated here, and the accuracy is found. And molecular screening and docking using MTI open screen and autodock, as screened open sources of ligand molecules and docked to find the bed fit orientation of the ligand. Ligplot to find the 2D representation of the protein ligand complexes. This is the structure of UMP CMP kinase that I have modeled, and this is the validation of Ramachandran plot of the protein structure. We can see that 98% are in the favored region and 2% is in the allowed region. This is a plot where the black dots are the amino acid residues, the darker blue region is a favored region. And the screening output here, we have screened nearly 1500 compounds, and we've obtained the ligands of which was the best bound of we obtained nearly 1110 that were very good binding. And this is the output of the 2D representation by Ligplot. We can see that the purple color structure here, the cyclic structure, is the ligand, and the other, we can see that the ligand and the protein bind to form a hydrogen bond at leucine 57A chain of the protein, and the ligand one. These are the other structures here are the hydrophobic interactions. The hydrophobic interactions are very important since we can adjust the interaction between them to form a perfect complex, which is more accurate and efficient. I would like to conclude by telling that since the Albugo candida species infect the plants in the growing stage and also in the early stage, the plants do not grow further, and they die at a very early stage without forming the fruits or the vegetables, and which results in the crop loss. And also when the infection forms white patches and abnormal growth, it is undesirable to be bought, and there is no profit for the farmers. And virtual screening and molecular docking are nowadays widely studied. Even in the COVID scenario, we are using molecular docking and screening to screen all the ligands from the library, ligand library, and we are docking them to find first the computational method, and then going for the wet lab techniques. So further research has to be done for confirming the inhibition of the kinase molecule with the protein. Thank you.